Welcome to Dreamy Facts. As the day gently recedes, we invite you to settle into a space of calm contemplation. Tonight, we embark on a journey through the human nervous system, exploring approximately 100 fascinating facts that reveal intricate patterns, subtle wisdom, and profound beauty within this extraordinary biological network. There is no pressure to remember or retain these details. Instead, let them unfold naturally, inviting your mind to settle into a state of peaceful curiosity. This exploration is designed to be a soothing experience, a gentle current of knowledge flowing without demanding effort or attention. We will observe the intricate dance of electrical impulses, the subtle adaptations of cellular structures, and the quiet wisdom embedded in the way our bodies perceive, react, and remember. Each fact is a small window into the vast and interconnected tapestry of the nervous system, presented in a calm and meditative tone, allowing your thoughts to drift and find stillness. Allow the gentle rhythm of these discoveries to guide you towards a state of relaxation, inviting a sense of wonder and peace as you prepare for rest. If you're finding peace in this journey, a like and subscribe helps us continue creating these restful explorations, and we're always curious. Share in the comments what city and time zone you're watching from. There's something special about knowing this experience is reaching people all over the world as they prepare for rest. As we gently begin our exploration, we turn our attention to the very core of our being, the human nervous system. This intricate biological marvel orchestrates every thought, every movement, every sensation, working tirelessly to connect us with the world and ourselves. Let us observe its foundational elements, understanding how this complex network is built from the ground up, directing the symphony of our existence. The human nervous system is an incredibly complex network, often described as the body's command and communication center. Research shows it is broadly divided into two main parts: the central nervous system, CNS, and the peripheral nervous system, PNS. The CNS comprises the brain and spinal cord, acting as the primary processing unit for all incoming information and outgoing commands. Scientists have observed that the brain alone contains an estimated 86 billion neurons, forming trillions of connections that allow for complex thought, emotion, and action. The spinal cord extending from the brainstem serves as a crucial information highway, transmitting signals between the brain and the rest of the body. This fundamental division allows for specialized functions, with the CNS handling high-level integration and the PNS acting as its vast communication network. At the heart of this intricate system are neurons, the fundamental building blocks responsible for transmitting electrical and chemical signals. Current understanding suggests that a typical neuron consists of three main parts: the cell body, soma, dendrites, and an axon. Dendrites are tree-like branches that receive incoming signals from other neurons, while the axon is a long, slender projection that transmits signals away from the cell body to other neurons, muscles, or glands. Studies indicate that there are several types of neurons, including sensory neurons, which carry information from sensory receptors to the CNS, motor neurons, which transmit signals from the CNS to muscles and glands, and interneurons, which connect neurons within the CNS. The ability of these specialized cells to generate and propagate electrical impulses, known as action potentials, is what enables rapid communication throughout the body. forming the basis of all nervous system functions. While neurons are often highlighted for their signal transmitting capabilities, they are far from alone in the nervous system. Glial cells, often referred to as neuroglia, play an equally vital, though often unsung, role. Scientists have observed that glial cells outnumber neurons in some parts of the brain, with some estimates suggesting a ratio of 1 in the human brain. These support cells perform a multitude of functions essential for neuronal health and function. For instance, oligodendrocytes in the CNS and Schwann cells in the PNS produce myelin, a fatty substance that insulates axons and significantly increases the speed of nerve impulse transmission. Astrocytes, another type of glial cell, provide structural support, regulate the chemical environment around neurons, and contribute to the blood-brain barrier. Microglia act as the immune cells of the CNS, protecting the brain from pathogens and clearing cellular debris. Without the tireless work of glial cells, the delicate network of neurons would quickly falter, highlighting their critical importance in maintaining the nervous system's integrity and efficiency. The speed at which nerve impulses travel through our body is truly remarkable, allowing for near instantaneous reactions and complex processing. Research shows that the fastest nerve impulses can travel at speeds exceeding 120 meters per second, or about 268 miles per hour. This incredible velocity is largely due to the presence of the myelin sheath, produced by glial cells, which acts as an electrical insulator around the axon. The impulse effectively jumps between gaps in the myelin, known as nodes of Ranvier, in a process called saltatory conduction, significantly accelerating transmission. In contrast, unmyelinated axons transmit impulses much more slowly, at speeds of around 0.5 to 10 meters per second. This variation in speed allows the nervous system to prioritize certain signals, such as those crucial for reflexes or immediate danger, ensuring that vital information reaches its destination with astonishing rapidity. At the junction between two neurons, or between a neuron and another cell, lies a specialized structure called a synapse. This is the crucial point where information is transferred. Current understanding suggests that synapses can be either electrical or chemical, though chemical synapses are far more common in the human nervous system. In a chemical synapse, when an electrical impulse, action potential, reaches the end of the transmitting neuron's axon, it triggers the release of chemical messengers called neurotransmitters into the synaptic cleft, a tiny gap between the two cells. These neurotransmitters then bind to specific receptors on the receiving cell, generating a new electrical signal or eliciting a response. Scientists have identified over 100 different neurotransmitters, each with specific roles, such as dopamine involved in reward and motivation, serotonin influencing mood, and acetylcholine crucial for muscle contraction. This intricate chemical communication allows for nuanced and highly regulated signaling throughout the nervous system. As we gently drift further into the intricate landscape of our inner workings, we find ourselves at the very heart of communication, where the whisper of one neuron sparks a cascade of profound understanding. The previous chapter introduced us to the fundamental architecture, the neurons, the glial cells, the rapid electrical impulses, and the synaptic junctions where messages leap across microscopic divides. Now we deepen our exploration. turning our gaze to the silent symphony conducted within these spaces, the world of neurotransmitters and the grand orchestrator that is our brain. We left off at the synapse, that remarkable meeting point where an electrical signal transforms into a chemical messenger. These messengers, the neurotransmitters, are the very language of our nervous system, dictating not just movement and sensation, but also the nuances of our thoughts, emotions, memories, and even the subtle shifts in our consciousness. They are the chemical keys unlocking a vast spectrum of human experience, each with its own unique role in the intricate dance of neural communication. Current understanding suggests there are over 100 known neurotransmitters, each contributing to the vast and interconnected tapestry of our inner world. Let us explore a few of these crucial players, appreciating their subtle wisdom and profound impact. Consider acetylcholine, ACH, often regarded as one of the first neurotransmitters discovered. Research indicates its vital role in muscle contraction, acting as the primary messenger between motor neurons and muscle fibers. But its influence extends far beyond movement. Studies have shown its critical involvement in learning, memory, and attention within the brain. A calm, focused state, a vivid memory recalled, or the simple act of lifting a hand, all carry the signature of acetylcholine's diligent work. Then there is dopamine, a neurotransmitter frequently associated with the brain's reward system. Scientists have observed that dopamine release is linked to feelings of pleasure, motivation, and reinforcement. It plays a significant part in our drive to seek out new experiences, to learn, and to achieve goals. But its role is far more complex than just feeling good. Dopamine pathways are also crucial for controlling movement, with disruptions leading to conditions like Parkinson's disease and influencing our decision-making processes. It is a powerful chemical, shaping our pursuits and our perceptions of reward. Serotonin. Another widely recognized neurotransmitter is often linked to mood regulation, sleep, appetite, and digestion. Its balanced presence contributes to feelings of well-being and calmness, while imbalances are frequently implicated in mood disorders. Imagine a gentle current flowing through your mind, bringing a sense of peace and contentment. That subtle wisdom is often attributed to the steady hand of serotonin. It helps us to regulate our internal rhythms, guiding us towards a state of relaxation and mental equilibrium. The nervous system also relies on a delicate balance of excitatory and inhibitory neurotransmitters. Glutamate is the primary excitatory neurotransmitter in the central nervous system, meaning it primarily drives neurons to fire, playing a critical role in learning and memory formation. Without glutamate, our brains would struggle to form new connections and store information. Conversely, gamma-aminobutyric acid, GABA, is the main inhibitory neurotransmitter, acting like a gentle brake on neural activity. It reduces neuronal excitability throughout the nervous system, promoting relaxation, reducing anxiety, and helping to maintain a calm, stable neural environment. The interplay between glutamate and GABA is a fundamental aspect of healthy brain function, ensuring that our neural circuits are neither overstimulated nor underactive. And let us not forget the endorphins, our body's natural painkillers and mood elevators. These remarkable chemicals are released in response to stress, pain, and even intense exercise, creating feelings of euphoria and well-being. They are the internal balm, soothing discomfort and allowing us to push through challenges, a profound testament to the nervous system's capacity for self-care. This intricate interplay of neurotransmitters, released and absorbed in milliseconds, forms the chemical ballet that underpins every aspect of our existence. It is a testament to the profound beauty of our internal landscape, a constant, silent symphony of signals that shapes who we are and how we experience the world. As we move beyond the microscopic dance of neurotransmitters, our peaceful curiosity leads us to the grand stage where these signals converge and are processed. The central nervous system, CNS. We have already noted its two main components, the brain and the spinal cord. Now we invite you to gaze upon the brain, the ultimate master controller, a marvel of biological engineering that continues to inspire awe and endless scientific inquiry. The human brain is an organ of astonishing complexity and capacity, weighing approximately 3 pounds in an adult, yet containing an estimated 86 billion neurons, each forming thousands of connections. This creates a network of staggering density and potential, allowing for consciousness, creativity, reasoning, emotion, and the intricate coordination of our entire being. It is the seat of our self, the engine of our thoughts, and the interpreter of our reality. We can broadly divide the brain into 3 main parts: the cerebrum, the cerebellum, and the brain stem. The cerebrum is the largest part of the brain, the wrinkled, outer layer that we most commonly associate with intelligence and higher level functions. It is divided into 2 hemispheres, the left and right, each specialized for certain tasks, yet constantly communicating through a thick band of nerve fibers called the corpus callosum. The cerebrum's surface, the cerebral cortex, is where our conscious thought, language, memory, and voluntary movements originate. It is a vast landscape of intricate patterns, folded into gyri, ridges, and sulci, grooves, maximizing its surface area to pack in an incredible amount of processing power. Within the cerebrum, scientists have identified distinct regions, or lobes, each with primary responsibilities. The frontal lobe, located at the front of the brain, is the command center for executive functions. Current understanding suggests it is responsible for planning, decision-making, problem-solving, voluntary movement, and personality. It is where we formulate our intentions and orchestrate complex behaviors, making us uniquely human. The parietal lobe, situated behind the frontal lobe, processes sensory information from the body, including touch, temperature, pain, and pressure. It also plays a crucial role in spatial awareness and navigation, allowing us to understand our position in the world and interact with objects around us. The temporal lobe, found on the sides of the brain, just above the ears, is essential for processing auditory information, understanding language, and forming memories. It houses the hippocampus, a structure vital for converting short-term memories into long-term ones, and the amygdala, central to processing emotions like fear. The occipital lobe, located at the very back of the brain, is dedicated almost entirely to processing visual information. It is where raw visual data from our eyes is interpreted, allowing us to perceive shapes, colors, and motion, transforming light into meaningful images. Beneath the grand expanse of the cerebrum, nestled at the back of the brain, lies the cerebellum, often referred to as the little brain. Despite its smaller size, it contains more neurons than the rest of the brain combined, playing an indispensable role in motor control, coordination, balance, and fine-tuning movements. From walking gracefully to playing a musical instrument, the cerebellum ensures our movements are smooth, precise, and well-timed. Research indicates it also contributes to cognitive functions, including language and attention, revealing its broader influence on our mental landscape. Connecting the cerebrum and cerebellum to the spinal cord is the brainstem, a vital stalk-like structure at the base of the brain. This ancient and essential part of our nervous system acts as a crucial relay station, transmitting signals between the brain and the rest of the body. More importantly, it regulates many of our most fundamental involuntary life-sustaining functions: breathing, heart rate, blood pressure, sleep, and consciousness. Without the brainstem, life as we know it would be impossible. It is the silent guardian, ceaselessly working to maintain the delicate balance required for our very existence. The brain's ability to adapt and reorganize itself throughout life is known as neuroplasticity. This profound beauty allows our brains to form new neural connections, strengthen existing ones, or even reassign functions to different areas in response to learning, experience, injury, or environmental changes. It means that our brain is not a static organ, but a dynamic, ever-evolving landscape, constantly shaped by our interactions with the world and our internal states. This capacity for change is a testament to the subtle wisdom embedded within our nervous system, offering hope and potential for growth and recovery. From the brain, our journey gently descends into the spinal cord, the other major component of the central nervous system. Encased within the protective column of our vertebrae, the spinal cord is far more than just a simple cable connecting the brain to the body. It is a vital communication highway and a processing center in its own right. The spinal cord extends from the brainstem down to the lower back, serving as the primary conduit for information flow. Sensory information from the body, touch, pain, temperature, proprioception, our sense of body position, travels up the spinal cord to the brain for interpretation. Conversely, motor commands from the brain travel down the spinal cord to instruct muscles and glands throughout the body. It is the crucial bridge, ensuring that the brain is constantly aware of our internal and external environments and can respond effectively. Beyond its role as a relay, the spinal cord also acts as a local processing center for reflex arcs. These are rapid, involuntary responses to stimuli that do not require input from the brain, allowing for incredibly fast reactions to protect the body from harm. Imagine touching a hot stove. The sensory information travels to the spinal cord, which immediately sends a motor command back to the muscles to withdraw your hand, all before the sensation of pain has even fully registered in your brain. This intricate dance of immediate response showcases the profound beauty of the nervous system's efficiency and protective mechanisms. As we bring our calm contemplation to a close for this chapter, we reflect on the stunning complexity we have uncovered. From the nuanced language of neurotransmitters shaping our every thought and feeling, to the magnificent architecture of the brain with its specialized lobes and vital control centers, and the essential communication highway of the spinal cord, each element works in concert, a vast and interconnected tapestry weaving the symphony of our existence. We invite you to carry this peaceful curiosity with you allowing these dreamy facts to settle gently within your mind. As you drift and find stillness, perhaps you can appreciate the silent, ceaseless work happening within you, orchestrating your very being. In our next chapter, we will venture further, exploring the vast network of the peripheral nervous system and how it connects this central command center to every corner of your remarkable body. Until then, may your contemplations be serene and your dreams filled with wonder. As we gently drift from the profound depths of the brain and spinal cord, the central command centers we explored in our last shared moment, we now turn our gaze outward, following the intricate pathways that branch and weave throughout the entirety of our being. We embark on a serene journey into the vast and wondrous expanse of the peripheral nervous system, or PNS, the body's eloquent messenger, the sprawling network that connects every whisper of sensation, every command of movement, every subtle shift in our internal landscape to the grand orchestrator within. Imagine, if you will, a magnificent tree. The central nervous system, with its brain and spinal cord, forms the mighty trunk and main branches, while the peripheral nervous system represents the countless smaller branches, twigs and leaves, reaching out to touch the light, to feel the wind, to draw sustenance from the earth. This intricate arborization ensures that no part of our existence remains isolated. Every cell, every tissue, every organ is brought into the symphony of our nervous system. We invite you to find stillness in this understanding, allowing the complexity to unfold with calm contemplation. The PNS is, in essence, the crucial bridge, the grand communication highway that carries information both to and from the central nervous system. It is composed of all the nerves that lie outside the brain and spinal cord, extending into our limbs, our organs, our skin, everywhere. Scientists have meticulously mapped these pathways, revealing a system of unparalleled efficiency and adaptability. Through the PNS, our inner world receives constant updates from the outer environment, and our inner wisdom, the directives from the brain, is carried to every muscle and gland, orchestrating our responses to the world and maintaining the delicate balance within. Within this expansive peripheral network, we discover two primary divisions, each serving distinct yet harmoniously integrated roles: the somatic nervous system and the autonomic nervous system. We shall now explore each with peaceful curiosity, unraveling their subtle wisdom. First, let us gently explore the somatic nervous system, SNS. This is the voluntary arm of our nervous system, the conscious controller that allows us to interact directly with our external world. It is through the SNS that we choose to lift a hand, to walk, to speak, to write. We perceive the warmth of the sun on our skin, the texture of a soft blanket, the gentle pressure of a touch, all thanks to the sensory neurons of the SNS. These specialized neurons act as vigilant sentinels, gathering information from our skin, muscles, and joints, transmitting these vital messages inward towards the spinal cord and brain. Research indicates that these sensory signals are crucial not only for our perception of the world, but also for our very sense of self in space. At the same time, the SNS contains motor neurons that carry commands from the CNS to our skeletal muscles. These are the muscles responsible for all our conscious movements, from the grand gestures of dance to the delicate precision of threading a needle. When you consciously decide to move, a signal originates in your brain, travels down the spinal cord, and then exits via a motor neuron in the PNS, which directly synapses with muscle fibers, causing them to contract. This direct line of communication is what grants us agency, allowing us to sculpt our environment and express our intentions. Current understanding highlights the remarkable speed and precision of this system, enabling instantaneous reactions and finely tuned motor skills. Beyond deliberate movement, the SNS also plays a crucial role in proprioception, our body's inherent awareness of its position and movement in space. Without looking, we know where our arms and legs are, whether we are standing or sitting, how much tension is in our muscles. This subtle, unconscious sense is mediated by specialized sensory receptors within our muscles, tendons, and joints, which constantly feed information back to the CNS via the PNS. It is a profound, often overlooked aspect of our existence, allowing for balance, coordination, and fluid motion, a testament to the nervous system's intricate patterns of self-monitoring. Now let us gently shift our focus to the autonomic nervous system, ANS, the involuntary maestro conducting the internal symphony of our body. Unlike the SNS, which responds to our conscious will, the ANS operates largely below the threshold of our awareness, tirelessly regulating the functions essential for life itself. We do not consciously command our heart to beat, our lungs to breathe, our digestion to proceed, or our pupils to dilate. These vital processes are orchestrated by the ANS, ensuring our survival and internal stability. Scientists often refer to this internal balance as homeostasis, and the ANS is its primary guardian. The ANS in turn is further divided into two complementary yet opposing branches, working in a beautiful dynamic equilibrium: the sympathetic nervous system and the parasympathetic nervous system. We invite you to observe their dance, one of activation and one of calm, reflecting the ebb and flow of our daily lives. The sympathetic nervous system is often described as our fight or flight response system. It is activated during times of stress, danger, or intense excitement, preparing the body to either confront a threat or rapidly escape from it. When the sympathetic system is engaged, a cascade of physiological changes occurs. Our heart rate accelerates, pumping blood more rapidly to our muscles. Our breathing deepens and quickens, increasing oxygen intake. Our pupils dilate, allowing more light to enter our eyes, sharpening our vision. Digestion slows or halts, diverting energy to more immediate needs. And our adrenal glands release hormones like adrenaline and norepinephrine, further heightening our alertness and energy levels. Research indicates that this ancient system is a powerful survival mechanism, finely tuned over millennia to respond to perceived threats. It is a burst of energy, a surge of readiness, preparing us for immediate action. In contrast, the best parasympathetic nervous system is our rest and digest system, the gentle current that guides us back to a state of relaxation and replenishment. It becomes active during periods of calm, facilitating processes that conserve energy and promote healing. When the parasympathetic system is dominant, our heart rate slows, our breathing becomes more regular and shallow, our pupils constrict, and digestive processes are stimulated, allowing our body to absorb nutrients and eliminate waste. This system promotes a sense of tranquility, encouraging the body to rebuild its reserves and return to a state of peaceful equilibrium. It is the soothing balm after exertion, the quiet hum of restoration, allowing us to drift and find stillness. The interplay between these two systems is a constant, subtle dance, ensuring that our internal environment is always optimally adapted to our external circumstances, a profound beauty in its intricate regulation. Beyond these fundamental divisions, the PNS comprises specific types of nerves that emerge from different parts of the CNS. We have the uncranial nerves, a unique set of 12 pairs that arise directly from the brain itself, primarily from the brainstem. Unlike spinal nerves, which primarily serve the torso and limbs, cranial nerves mostly innervate structures in the head and neck, though one, the vagus nerve, extends its influence widely to organs in the chest and abdomen. These cranial nerves are fascinating in their diversity. For instance, the olfactory nerve, cranial nerve Y, is purely sensory, responsible for our sense of smell, allowing us to detect the subtle fragrances of the world. The optic nerve, cranial nerve 2, also purely sensory, transmits visual information from our eyes to our brain, painting the vibrant tapestry of our sight. Others, like the oculomotor, cranial nerve 3, trochlear, cranial nerve GAND, and abducens, cranial nerve 6 nerves, are primarily motor, controlling the delicate movements of our eyeballs. The trigeminal nerve, cranial nerve ver, is a mixed nerve, providing sensation to the face and controlling muscles for chewing. The facial nerve, cranial nerve 7, controls facial expressions and our sense of taste. The vestibulocochlear nerve, cranial nerve 8, brings us the wonders of hearing and balance. The glossopharyngeal nerve, cranial nerve 9, is involved in taste, swallowing, and salivary gland control. The vagus nerve, cranial nerve X, a truly remarkable nerve, extends its reach far beyond the head, influencing our heart rate, digestion, and even our vocal cords, a profound testament to the interconnectedness of our systems. The accessory nerve, cranial nerve 11, controls neck and shoulder muscles, and the hypoglossal nerve, cranial nerve 2, controls tongue movements. Each cranial nerve is a specialized conduit, contributing to the rich sensory experiences and motor capabilities that define our interaction with the world. Complementing the cranial nerves are the spinal nerves, 31 pairs that emerge from the spinal cord at different levels, extending their tendrils throughout the rest of the body. Each spinal nerve is a mixed nerve, containing both sensory, afferent fibers that carry information to the spinal cord and motor, efferent fibers that carry commands from the spinal cord to muscles and glands. They are organized segmentally, meaning that specific spinal nerves generally innervate particular regions of the body, creating predictable patterns known as dermatomes, areas of skin supplied by a single spinal nerve, and myotomes, groups of muscles supplied by a single spinal nerve. As these spinal nerves exit the spinal cord, they often merge and redistribute in complex networks called suplexuses, before branching out to their final destinations. For example, the brachial plexus in the shoulder region gives rise to the nerves that supply our entire arm and hand, enabling the incredible dexterity and strength of our upper limbs. Similarly, the lumbar and sacral plexuses in the lower back contribute to the innervation of our legs and feet, facilitating movement and sensation. This intricate rerouting ensures that multiple spinal cord segments contribute to the function of a single limb, providing redundancy and robustness to our nervous system, a true marvel of biological engineering. The PNS is also home to an astonishing array of sensory receptors, specialized structures that act as transducers, converting various forms of energy from our environment or internal body into electrical signals that our nervous system can understand. These receptors are our primary interface with the world, our windows of perception. Consider the CERS mechanoreceptors, delicate sensors that respond to mechanical forces such as touch, pressure, vibration, and stretch. These are abundant in our skin, allowing us to feel the gentle caress of a breeze or the firm grip of a hand. Within our muscles and joints, specialized mechanoreceptors contribute to proprioception, providing constant feedback on our body's position. Then there are thermoreceptors, which detect changes in temperature, alerting us to warmth or cold, protecting us from extremes. The nociceptors are specialized pain receptors, vital for our survival, signaling potential tissue damage and prompting us to withdraw from harm. These are widely distributed throughout the body, acting as an essential warning system. Furthermore, we possess chemoreceptors, which respond to chemical stimuli. These are responsible for our senses of taste and smell, allowing us to savor flavors and detect scents. Internally, chemoreceptors monitor the chemical composition of our blood, detecting levels of oxygen, carbon dioxide, and pH, playing a crucial role in regulating breathing and maintaining our body's internal chemical balance. And while photoreceptors, which detect light, are primarily located in the retina of the eye, it is through the optic nerve, a component of the PNS, that their information is relayed to the brain, allowing us to perceive the vibrant spectrum of light and color that surrounds us. Each receptor type is a tiny, sophisticated instrument, translating the world into the language of neural impulses. Building upon our earlier discussion of reflex arcs, it is the PNS that forms the initial and final legs of these rapid involuntary responses. When you accidentally touch a hot surface, the sensory neurons of the PNS, specifically nociceptors, detect the heat and pain. This signal travels along a peripheral nerve to the spinal cord. Within the spinal cord, without necessarily involving the brain for the immediate response, the sensory neuron synapses directly or via an interneuron with a motor neuron of the PNS. This motor neuron then immediately sends a command back along a peripheral nerve to the muscles in your hand and arm, causing you to withdraw your hand almost instantaneously. This protective circuit, relying heavily on PNS components, highlights the incredible efficiency and speed with which our nervous system protects us from harm. It is a testament to the system's design for immediate life-preserving action. Finally, we briefly touch upon the chemical messengers within their PNS. Just as in the CNS, neurotransmitters facilitate communication at the synapses. At the neuromuscular junction, where motor neurons of the SNS meet skeletal muscle fibers, acetylcholine is the primary neurotransmitter, causing muscle contraction. Within the ANS, acetylcholine is also crucial, mediating communication within the parasympathetic nervous system, promoting its effects. Conversely, norepinephrine is the main neurotransmitter released by most postganglionic sympathetic neurons, driving the responses we discussed earlier. These chemical signals ensure the seamless and precise execution of both voluntary and involuntary commands throughout our vast peripheral network. As we conclude this exploration of the peripheral nervous system, we hope you feel a renewed sense of wonder for the intricate patterns and profound beauty within you. From the conscious commands that move your limbs to the silent, tireless regulation of your internal organs, the PNS is a vast and interconnected tapestry, constantly gathering information, relaying commands, and maintaining the delicate balance of your existence. It is a system of subtle wisdom, always at work, ensuring your harmony with the world and within yourself. We invite you to carry this calm contemplation forward, allowing the gentle current of discovery to deepen your appreciation for the symphony of our being. Find stillness now, and let these dreamy facts gently recede as you prepare for peaceful rest. As the gentle current of our exploration carries us forward, we now turn our gaze from the intricate pathways of the peripheral nervous system, those diligent messengers, towards the magnificent central processing unit that interprets their every signal. Having understood how the body gathers information and responds, we are ready to delve into the profound artistry of how our brain transforms raw data into the rich, vibrant tapestry of our conscious experience. We invite you to drift deeper into a state of calm contemplation, allowing the subtle wisdom of your own nervous system to unfold before your mind's eye. Imagine the vast ocean of incoming sensory information, a constant deluge of light, sound, touch, taste, and smell. It is here, within the quiet hum of your brain, that these disparate threads are woven into the coherent, meaningful reality we perceive. Our journey begins with the fascinating process of sensory integration and perception. It is a common misconception that our brain merely records reality as a camera or microphone might. On the contrary, research reveals that our brain is an active constructor of reality, constantly interpreting, predicting, and filling in gaps to create a seamless, personal narrative of the world around us. This intricate dance of interpretation is one of the most remarkable aspects of our neural existence. Consider, for instance, the thalamus, often referred to as the brain's grand relay station. Nestled deep within the cerebrum, this egg-shaped structure acts as a vital switchboard for almost all sensory information. Every sight, sound, touch, and taste signal, with the intriguing exception of smell, which takes a more direct route, first passes through the thalamus. Here, these signals are filtered, organized, and then meticulously routed to the appropriate specialized cortical areas for further processing. It is as if the thalamus is the conductor of an orchestra, ensuring each section receives its score at precisely the right moment, ready to contribute to the grand symphony of perception. Once past the thalamus, these signals arrive at their designated cortical destinations. For touch, temperature, pain, and proprioception, our sense of body position, the signals are sent to the somatosensory cortex, located in the parietal lobe. Here, a detailed map of the body exists, with certain areas, like the hands and face, receiving disproportionately larger representation due to their heightened sensitivity. This is where we consciously register the warmth of a gentle breeze, the texture of a soft blanket, or the subtle pressure of our own weight. Scientists have observed that this area not only processes the physical sensation, but also contributes to our awareness of our body's spatial relationship with its surroundings. The world of sight unfolds as light hits the retina, transforming into electrical impulses that travel along the optic nerve. This visual information makes its way to the thalamus, specifically the lateral geniculate nucleus, LGN, before being projected to the visual cortex in the occipital lobe at the very back of our brain. Here, an astonishing array of specialized neurons spring into action. Some neurons are dedicated to detecting edges and lines, others to specific colors, and still others to motion. Our perception of a vibrant sunset, the intricate patterns of a leaf, or the fleeting glimpse of a bird in flight is the culmination of these millions of neurons working in concert, meticulously dissecting and reassembling the visual world. Current understanding suggests that different pathways within the visual cortex process what an object is, its form and color, and where it is, its location and motion, allowing for a comprehensive understanding of our visual environment. Similarly, the intricate world of sound is processed in the auditory cortex, located within the temporal lobe. Sound waves are converted into neural signals in the cochlea of the inner ear, then travel via the auditory nerve, through the thalamus, specifically the medial geniculate nucleus MGN, and finally to the auditory cortex. Here, the brain deciphers the pitch, loudness, and timbre of sounds, enabling us to distinguish between a whispered word, a soaring melody, or the distant rumble of thunder. Research indicates that specific regions within the auditory cortex are responsible for processing different frequencies, allowing us to perceive the full spectrum of acoustic information and even localize the source of a sound in space. The sense of smell or olfaction offers a unique pathway, bypassing the thalamus and projecting directly to the olfactory bulb, which then sends signals to the primary olfactory cortex and directly into parts of the limbic system, including the amygdala and hippocampus. This direct connection explains why certain scents can evoke such powerful memories and emotions, sometimes without us even consciously realizing why. The fragrance of a familiar flower, the scent of rain on dry earth, or the aroma of a home-cooked meal can instantly transport us through time and space, a testament to the profound neural links between smell, memory, and emotion. Taste, or gustation, begins with our taste buds detecting sweet, sour, salty, bitter, and umami flavors. These signals are transmitted via several cranial nerves to the brainstem, then to the thalamus, and finally to the gustatory cortex, nestled deep within the insular and frontal operculum. Here, the brain integrates taste information with smell, texture, and temperature to create the complex and nuanced experience we call flavor. This multisensory integration highlights how our perceptions are rarely isolated, but rather a rich blend of concurrent sensory inputs. Indeed, multisensory integration is a cornerstone of our perceptual experience. Our brain constantly combines information from different senses to form a coherent, unified understanding of the world. Think of watching a movie. The visual and auditory information are seamlessly integrated, allowing us to perceive the dialogue as coming directly from the actor's mouth, even if the sound is projected from speakers to the side. This dynamic interplay between our senses enriches our perception, making it far more robust and adaptable than any single sense could achieve alone. Furthermore, Our nervous system exhibits a remarkable ability known as perceptual constancy. Despite constant changes in sensory input, as we move, as lighting shifts, as sounds vary in intensity, our brain strives to perceive objects and events as stable and unchanging. A door, for example, is still perceived as a rectangle, even as its retinal image distorts into a trapezoid when viewed from an angle. A person's voice maintains its identity whether they whisper or shout. This constancy allows us to navigate a dynamic world with a sense of stability and predictability. Underpinning all these sensory processes is the crucial function of REM, attention. In a world brimming with stimuli, our brain has developed sophisticated mechanisms to selectively focus on relevant information while filtering out the extraneous. This selective attention allows us to concentrate on a single conversation in a crowded room, or to spot a specific pattern amidst a complex background. Scientists understand that attention is not a single process, but involves a network of brain regions that modulate sensory processing, enhancing the signals we deem important and suppressing those we don't. It is the spotlight of consciousness, illuminating what matters most in any given moment. As we gently shift our focus, we move from the intricate symphony of sensation to the equally complex art of motor control. Our ability to move, from the slightest twitch of a finger to the most complex athletic feat, is a testament to the coordinated efforts of multiple brain regions working in exquisite harmony. This voluntary movement is not simply a reaction, but often a carefully planned and executed sequence of neural commands. The journey of a voluntary movement begins in the higher centers of the brain, specifically the prefrontal cortex, premotor cortex, and supplementary motor area. These regions are involved in the planning, decision-making, and sequencing of movements. Before you even lift a hand, these areas are engaged in considering the goal, the context, and the optimal strategy for action. It's like the architectural phase of building, where every detail is meticulously designed before construction begins. Once a movement plan is formulated, it is sent to the primary motor cortex, located in the frontal lobe, just in front of the somatosensory cortex. This is the executive command center for initiating specific voluntary movements. Different parts of the primary motor cortex control different parts of the body, forming a motor homunculus, a distorted map reflecting the fine motor control exerted over areas like the hands, face, and tongue, which require a larger neural representation. The commands for movement then travel down the spinal cord via the corticospinal tract, a major descending pathway. These neural fibers synapse with motor neurons in the spinal cord, which in turn send signals directly to the muscles, causing them to contract and produce movement. This direct communication ensures rapid and precise execution of our intentions. However, the process is far from a simple one-way street. Two other critical structures play vital roles in refining and coordinating movement: the basal ganglia and the cerebellum. The basal ganglia, a group of interconnected nuclei deep within the cerebrum, act like a sophisticated filter, facilitating desired movements while inhibiting unwanted ones. They are crucial for initiating movements, smoothing out actions, and learning new motor skills. Disruptions in the basal ganglia, as seen in conditions like Parkinson's disease, vividly illustrate their importance in allowing us to move freely and fluidly. The cerebellum, which we touched upon in a previous chapter, further refines movement. It continuously compares the intended movement with the actual movement, using sensory feedback to correct any discrepancies. This allows for precise coordination, balance, and the smooth, graceful execution of complex motor sequences. The cerebellum is also intimately involved in motor learning, enabling us to acquire new skills, from riding a bicycle to playing a musical instrument, through repeated practice and refinement. Each repetition strengthens the neural pathways, making the movement more automatic and efficient. Indeed, feedback loops are integral to motor control. As we move, our sensory receptors, particularly proprioceptors in our muscles and joints, constantly send information back to the brain about our body's position and the ongoing movement. This real-time sensory input allows the brain to make continuous adjustments, ensuring that our actions are accurate and adaptive to changing circumstances. It is a continuous conversation between intention and execution, guided by sensory awareness. Finally, we turn our attention to the neural underpinnings of our inner world, the fascinating realm of emotions and memory. These profound aspects of human experience are deeply intertwined with specific brain structures, primarily forming what scientists refer to as the limbic system. This ancient and powerful network plays a central role in shaping who we are, how we feel, and what we remember. At the heart of the limbic system lies the amygdala, a pair of almond-shaped structures crucial for processing emotions, particularly fear, anger, and pleasure. Research indicates that the amygdala is central to our fight or flight responses, rapidly evaluating potential threats and orchestrating our body's reaction. It also plays a significant role in emotional learning, helping us associate certain stimuli with emotional outcomes, a process known as fear conditioning. The amygdala imbues our experiences with emotional significance, coloring our memories and guiding our responses. Adjacent to the amygdala, and equally vital, is the hippocampus, another key structure within the limbic system shaped somewhat like a seahorse. The hippocampus is absolutely critical for the formation of new, explicit memories. Those conscious recollections of facts, events, and experiences. Without a functioning hippocampus, individuals struggle to form new declarative memories, living in a perpetual present. It is here that we consolidate the details of our day, the names of new acquaintances, and the information we learn. Scientists distinguish between different types of memory. Explicit or declarative memory includes episodic memory, recollections of specific personal events like your last birthday, and semantic memory, general knowledge and facts like the capital of France. In contrast, implicit or non-declarative memory operates largely unconsciously. This includes procedural memory, skills and habits like riding a bike or playing an instrument, and memories formed through classical conditioning, like salivating at the smell of food. While the hippocampus is vital for explicit memory, other brain regions such as the basal ganglia and cerebellum are more involved in implicit memory. The process of moving short-term memories into more permanent long-term storage is known as memory consolidation. and studies indicate that this process is significantly enhanced during sleep. During deep sleep, the brain appears to replay and strengthen neural connections associated with recent learning, solidifying our experiences and knowledge. The prefrontal cortex, the most anterior part of the frontal lobe, also plays a sophisticated role in emotional regulation, working memory, and decision-making. It allows us to modulate our emotional responses, to plan for the future, and to hold information in mind for short periods while we work with it. It is the seat of executive function, allowing for complex thought and behavioral control. And of course, the orchestra of neurotransmitters continues to play a pivotal role in these higher functions. Dopamine, beyond its role in movement, is intricately linked to reward and motivation, influencing our emotional responses and the formation of memories associated with pleasurable experiences. Serotonin profoundly impacts mood, anxiety, and learning, while acetylcholine is critical for attention, learning, and memory, particularly in the hippocampus. The delicate balance of these chemical messengers profoundly shapes our emotional landscape and our ability to learn and recall. As we gently recede from this chapter, we hope you feel a deepened sense of wonder for the intricate mechanisms that allow you to perceive, move, feel, and remember. The symphony of sensation, the art of movement, and the profound depths of emotion and memory are not abstract concepts, but the very fabric of your being, orchestrated by the subtle wisdom of your nervous system. In our next shared moment of peaceful curiosity, we will continue our exploration into even more complex aspects of this magnificent biological marvel, inviting you to drift and find stillness within the knowledge of yourself. As the gentle current of our exploration carries us deeper into the intricate patterns of the human nervous system, we find ourselves drifting from the foundational mechanics of memory and emotion gracefully into the expansive realm of higher thought. We invite you now to settle into a state of calm contemplation, allowing your peaceful curiosity to guide us through the fascinating architectures that underpin our unique capacity for language, complex decision-making, and the very rhythms that orchestrate our daily existence. This journey promises to illuminate the profound beauty of the brain's most sophisticated operations, revealing how these delicate neural networks weave the rich tapestry of our consciousness. Our ability to communicate, to share ideas, stories, and emotions through the intricate dance of language stands as one of the nervous system's most remarkable achievements. It is a symphony of coordinated neural activity, far more complex than a simple output of sounds or input of words. Language processing involves specific interconnected brain regions. Research has consistently shown that our capacity for language is not diffuse, but rather localized to specialized areas within the cerebral cortex, primarily in what is often referred to as the language network. These regions work in concert, each contributing a unique facet to the overall linguistic experience. We witness here a testament to the brain's organizational genius, where distinct functions are assigned to precise neural territories, yet remain deeply integrated. Broca's area is primarily responsible for speech production. Nestled in the frontal lobe, typically on the left side, Broca's area is a crucial hub for the motor aspects of speech. Scientists have observed that damage to this region can lead to, quote, 11. a condition where individuals struggle to produce fluent speech, often speaking in short, fragmented sentences, even though their comprehension might remain largely intact. It is here that the abstract thought of a word is translated into the precise muscular commands required to articulate it. Wernicke's area is critical for language comprehension. Further back in the temporal lobe, Wernicke's area serves as the primary processing center for understanding spoken and written language. When this area is affected, individuals may experience Quote 12, characterized by fluent but often nonsensical speech, coupled with significant difficulty in understanding language. They may speak in long sentences that lack meaning, unaware of their communication deficit. This highlights the distinct neural pathways for understanding and producing language. The arcuate fasciculus connects Broca's and Wernicke's areas. This vital bundle of nerve fibers acts as a superhighway, ensuring seamless communication between the speech production and comprehension centers. Current understanding suggests that the integrity of the arcuate fasciculus is essential for fluent, coherent speech, allowing us to not only understand what we hear but also to formulate appropriate responses. Damage to this pathway can lead to, quote 13, where individuals can comprehend and produce speech, but struggle to repeat words or phrases. Language is not solely processed in the dominant hemisphere. Prosody and context are also vital. While Broca's and Wernicke's areas are typically in the left hemisphere for most individuals, the right hemisphere plays a significant role in processing the emotional nuances of language, known as prosody. This includes the intonation, rhythm, and stress in speech that convey sarcasm, anger, or joy. Studies indicate that comprehending metaphors, humor, and the overall contextual meaning of language also heavily relies on right hemisphere activity, painting a richer, more holistic picture of how our brains experience communication. Bilingualism can lead to distinct neural pathways and cognitive advantages. For those who navigate 2 or more languages, the nervous system adapts with remarkable flexibility. Research suggests that bilingual individuals often exhibit increased grey matter density in certain brain regions and enhanced executive functions, such as attention shifting and task switching. The constant mental toggling between languages appears to strengthen cognitive control networks, offering a powerful example of experience-dependent neuroplasticity in action. Beyond the elegance of language, our nervous system empowers us with the profound ability to make decisions, to plan for the future, and to exert control over our impulses. These, quote 14, represent the pinnacle of cognitive processing, allowing us to navigate the complexities of our world with purpose and foresight. The prefrontal cortex is the seat of executive functions. As we journey deeper into the frontal lobe, we encounter the prefrontal cortex, the brain's ultimate command center for higher-order thinking. This region is instrumental in planning, problem-solving, working memory, impulse control, and the intricate process of decision-making. It is here that we weigh options, predict consequences, and regulate our behavior to achieve long-term goals, distinguishing us as truly complex beings. Decision-making often involves a delicate interplay between reason and emotion. While we might perceive our decisions as purely rational, scientists have observed that emotional input from the limbic system significantly influences our choices. The nervous system doesn't operate in isolated compartments. Rather, the prefrontal cortex integrates emotional signals, particularly from the amygdala, to guide us towards decisions that are not only logical but also emotionally resonant or personally meaningful. This integration helps us evaluate the subjective value of potential outcomes. The ventral medial prefrontal cortex, VMPFC, is crucial for integrating emotion into rational decision-making. Studies involving individuals with damage to the VMPFC demonstrate a fascinating insight. While their logical reasoning may remain intact, they often struggle with making advantageous decisions in real-life scenarios, particularly those involving risk and reward. This suggests that the VMPFC plays a pivotal role in linking emotional states with cognitive evaluations, allowing us to, quote, "15 our way towards better choices rather than relying solely on cold logic." Cognitive biases reveal how our nervous system often takes shortcuts in decision-making. Our brains are remarkably efficient, but this efficiency sometimes comes at the cost of perfect rationality. Research in cognitive psychology reveals numerous biases, systematic errors in thinking that influence our decisions. From confirmation bias, seeking information that confirms existing beliefs, to anchoring bias, over-relying on the first piece of information offered, these shortcuts illustrate how the nervous system, in its effort to conserve energy and process information quickly, can sometimes lead us astray. Working memory, a temporary storage system, is crucial for complex thought and decision processes. Distinct from long-term memory, working memory allows us to hold and manipulate information in our minds for short periods, enabling us to follow instructions, solve mental arithmetic problems, or keep track of the steps in a complex task. This capacity, largely attributed to the prefrontal cortex, is indispensable for planning, reasoning, and making informed decisions in real time, acting as a mental scratchpad for our conscious thoughts. As our conscious exploration gently recedes, we prepare to embrace another fundamental aspect of our nervous system, the profound and restorative cycles of sleep. Far from being a state of passive rest, sleep is an active, neurologically orchestrated process, deeply intertwined with our overall well-being and cognitive function. Sleep is an active, vital neurological process, not merely a state of inactivity. This gentle truth reminds us that even as our bodies rest, our brains are diligently working. During sleep, the nervous system engages in a complex array of activities crucial for consolidation of memories, emotional regulation, and the physiological maintenance of brain health. It is a period of intense internal processing, essential for both our mental clarity and physical restoration. The suprachiasmatic nucleus, SCN, in the hypothalamus acts as the body's master clock. Deep within the hypothalamus, a tiny cluster of neurons known as the suprachiasmatic nucleus orchestrates our circadian rhythms, the approximately 24-hour cycles that govern our sleep-wake patterns, hormone release, and many other physiological processes. This internal clock is exquisitely sensitive to light cues, primarily received through specialized photoreceptors in the retina, ensuring our internal rhythms remain synchronized with the external world. Melatonin, produced by the pineal gland, helps regulate sleep-wake cycles. As darkness falls, the SCN signals the pineal gland, a small endocrine gland in the brain, to release melatonin, This hormone acts as a chemical signal for night, promoting feelings of drowsiness and preparing the body for sleep. Conversely, light exposure inhibits melatonin production, reinforcing wakefulness. This elegant system underscores the nervous system's sophisticated control over our daily rhythms. Sleep consists of distinct stages: NREM, N1, N2, N3, slow wave sleep, and REM sleep. Our journey through the night is not uniform but a cyclical progression through different states of consciousness. Non-rapid eye movement NREM sleep is characterized by decreasing brain activity and muscle relaxation, deepening from light sleep N1 to deeper sleep N2, and finally to slow wave sleep N3, the most restorative phase. Rapid eye movement, REM sleep, in contrast, is marked by high brain activity, vivid dreaming, and temporary muscle paralysis. NREM sleep is crucial for physical restoration and declarative memory consolidation. During the profound stillness of NREM sleep, particularly slow wave sleep, our bodies repair and restore themselves. Simultaneously, the brain actively consolidates declarative memories, facts, events, and personal experiences, transferring them from temporary storage in the hippocampus to more permanent storage in the cortex. This is where the day's learning becomes etched into our long-term knowledge. REM sleep is associated with dreaming, emotional processing, and procedural memory consolidation. As we transition into REM sleep, our brains become remarkably active, often generating vivid dreams. This stage is vital for emotional regulation, helping us process the day's experiences and integrate them into our emotional landscape. Furthermore, REM sleep plays a significant role in consolidating procedural memories, skills, habits, and unconscious knowledge, allowing us to refine abilities like playing an instrument or riding a bike. During sleep, the glymphatic system actively clears metabolic waste products from the brain. A truly remarkable discovery, the glymphatic system, a network of glial cells, acts as the brain's unique waste removal system. Research indicates that during sleep, the channels of this system expand, allowing cerebrospinal fluid to flush out harmful metabolic byproducts, including amyloid beta proteins implicated in neurodegenerative diseases. This nightly cleansing ritual highlights the profound restorative power of sleep for long-term brain health. Our exploration of the nervous system would be incomplete without a deeper appreciation for its most astonishing quality, its capacity for change, adaptation, and growth. This phenomenon, known as neuroplasticity, allows our brains to be sculpted by experience, learning, and even recovery from injury. Learning fundamentally involves changes in synaptic strength and the formation of new connections. Every new skill acquired, every new piece of information absorbed, reshapes the very architecture of our brains. At the microscopic level, learning involves synaptic plasticity, the strengthening or weakening of connections between neurons, synapses, and even the formation of entirely new synaptic connections. This constant rewiring is the biological basis of memory and adaptation. Long-term potentiation, LTP, is a persistent strengthening of synapses, a key mechanism for learning. Scientists have identified specific molecular mechanisms that underpin this synaptic strengthening. Long-term potentiation is a process where a prolonged increase in synaptic strength occurs between two neurons that are repeatedly activated together. This neurons that fire together, wire together principle makes the transmission of signals between these neurons more efficient and enduring, forming the cellular foundation for learning and memory formation. Experience-dependent plasticity allows the brain to reorganize and adapt throughout life. From mastering a new language to navigating a complex new environment, our brains are continuously reorganizing in response to our experiences. This form of plasticity is not limited to childhood but persists throughout the lifespan, enabling adults to learn new skills, adapt to sensory changes, and even recover some function after brain injury. The brain is not a static organ but a dynamic, ever-evolving landscape. Skill acquisition leads to measurable changes in motor cortex representation. When you learn to play a musical instrument or master a sport, the areas of your motor cortex dedicated to controlling the relevant body parts actually expand and refine their neural connections. This is a vivid example of how focused practice and repeated experience can literally remap brain regions, making movements more precise, efficient, and automatic. Environmental enrichment can enhance neurogenesis in certain brain areas. While once thought to be limited to development, research has revealed that new neurons, neurogenesis, can be born in specific regions of the adult brain, most notably the hippocampus. Studies indicate that enriched environments, those offering novel experiences, physical activity and social interaction, can stimulate this process, potentially boosting cognitive function and resilience. This suggests that engaging with a stimulating world literally helps our brains grow. As we gently bring this chapter to a close, we hope you feel a renewed sense of wonder for the extraordinary capabilities housed within your nervous system. From the intricate dance of language that allows us to connect, to the thoughtful processes of decision-making and the vital restorative rhythms of sleep, each aspect contributes to the profound and complex symphony of our existence. Allow these facts to settle into your mind, fostering a deeper appreciation for the subtle wisdom that guides you, day and night. In our next shared moment of peaceful curiosity, we will continue our journey, exploring even more intricate dimensions of this magnificent system. As the gentle current of our exploration carries us deeper into the wondrous intricacies of the human nervous system, we find ourselves ever more captivated by its profound adaptability and its ceaseless dance with the world, both internal and external. We have journeyed through the foundational structures, the symphony of neurotransmitters, the vast network of the peripheral system, and the higher faculties of perception, memory, and cognition. Now, we invite you to drift with us into a contemplation of its remarkable resilience, its hidden connections, and its pathways to renewal. We understand that our nervous system is not a static entity, but a dynamic, living tapestry, constantly responding, learning, and striving for balance. In this chapter, we turn our gaze toward how this intricate system navigates the challenges of daily life, particularly stress, and how it communicates with seemingly distant parts of our being, revealing an interconnectedness more profound than we might first imagine. We will also explore the subtle forces that can disturb its harmony and, crucially, its inherent capacity for healing and adaptation. In our previous explorations, we touched upon the autonomic nervous system, that silent orchestrator of our internal world, with its sympathetic branch rousing us to action, and its parasympathetic branch inviting us to rest. Yet, the nervous system's response to challenge extends far beyond these immediate reactions, intertwining with our endocrine system in a complex and vital partnership known as the hypothalamic pituitary adrenal HPA axis. This intricate neuroendocrine pathway is our body's central stress response system, a finely tuned mechanism designed to help us adapt and survive. Imagine a subtle alarm bell ringing deep within the brain's emotional core. This signal, often triggered by a perceived threat or challenge, reaches the hypothalamus, a pea-sized region nestled at the base of the brain which acts as the HPA axis's control center. In response, the hypothalamus releases a hormone called corticotropin-releasing hormone, CRH. This CRH then travels a short distance to the pituitary gland, often referred to as the master gland of the endocrine system, where it stimulates the release of adrenocorticotropic hormone, ACTH. From there, ACTH embarks on a journey through the bloodstream, reaching the adrenal glands, which sit atop our kidneys. These small, powerful glands, upon receiving the ACTH signal, spring into action, releasing a cascade of stress hormones, most notably cortisol and adrenaline, epinephrine. Adrenaline provides that immediate surge of energy, sharpening our senses and preparing us for fight or flight, a rapid response we explored earlier. Cortisol, however, plays a more sustained and multifaceted role. Research reveals that cortisol mobilizes energy reserves by increasing glucose in the bloodstream, suppresses non-essential bodily functions like digestion and the immune system, and modulates brain activity. It helps us focus on the immediate threat, enhancing memory retrieval for stress-related events, and even temporarily boosting certain cognitive functions. This acute stress response is profoundly adaptive, allowing us to react swiftly and effectively to dangers. However, current understanding suggests that when this HPA axis remains chronically activated, due to ongoing stressors like work pressure, relationship difficulties, or persistent worry, the adaptive benefits begin to wane, giving way to detrimental effects. Prolonged exposure to high levels of cortisol can start to reshape the very structures of our brain. Studies indicate that chronic stress can lead to a reduction in the volume of the hippocampus, a brain region critical for memory and learning, and can impair the function of the prefrontal cortex, our center for executive functions, decision-making, and emotional regulation. This can manifest as difficulties with memory, increased anxiety, and even contribute to mood disorders. Scientists have observed that the nervous system strives for allostasis, a process of achieving stability through change. While homeostasis maintains a fixed set point, allostasis involves constantly adjusting physiological parameters to meet ongoing demands. When the demands are too great or too prolonged, the system can become dysregulated, leading to an allostatic load that wears down the body and mind, highlighting the profound importance of finding moments of calm and allowing our nervous system to gently recalibrate. As we contemplate the vast interconnectedness of our internal world, we are increasingly drawn to a fascinating and relatively newly appreciated dialogue that occurs far from the cranium, yet profoundly influences our mental landscape, the gut-brain axis. This bidirectional communication system links the central nervous system, CNS, with the enteric nervous system, ENS, often referred to as our second brain, residing within the walls of our digestive tract. The ENS is an astonishing network, comprising hundreds of millions of neurons embedded in the gut lining, capable of operating independently to manage the complex processes of digestion, from swallowing to nutrient absorption to waste elimination. Yet, it is far from isolated. This second brain is in constant, intricate communication with our primary brain, a whispering highway of signals and chemical messengers. This communication flows in both directions, creating a delicate feedback loop. From the brain to the gut, stress, as we just discussed, can profoundly impact gut motility, secretion, and even the composition of our gut microbiome. Have you ever felt butterflies in your stomach when nervous? That's your brain sending signals down to your gut. More remarkably, the gut also communicates with the brain, influencing our mood, emotions, and even cognitive functions. One primary pathway for this is the vagus nerve, a major cranial nerve that acts as a superhighway, carrying signals directly from the gut to the brainstem and beyond. But the communication doesn't stop there. Current research illuminates the critical role of the gut microbiome, the trillions of bacteria, fungi, and other microorganisms residing in our intestines. These microscopic inhabitants are not mere passengers, they are active participants in our neurochemical symphony. Scientists have discovered that gut microbes can produce a wide array of neuroactive compounds, including neurotransmitters like serotonin and GABA, which are identical to those produced in the brain. Indeed, a significant portion of the body's serotonin, a key regulator of mood and well-being, is synthesized in the gut. These microbial-produced neurotransmitters, along with other metabolites and immune signals, can influence brain function, modulating our stress response, anxiety levels, and even our social behaviors. An imbalance in the gut microbiome, known as dysbiosis, has been linked in studies to various neurological and psychiatric conditions, from anxiety and depression to autism spectrum disorder and Parkinson's disease, highlighting the profound impact of our internal ecosystem on our mental state. Furthermore, the integrity of the gut lining plays a crucial role. A compromised or leaky gut can allow inflammatory molecules to enter the bloodstream, potentially crossing the blood-brain barrier and contributing to systemic inflammation, which can in turn affect brain health and function. This intricate dance between our gut and our brain underscores the holistic nature of our well-being, reminding us that true health resonates throughout our entire being. As we gently expand our understanding of the nervous system's internal landscape, we encounter a phenomenon that is both protective and, when dysregulated, potentially destructive. Neuroinflammation. In the previous, we introduced glial cells, the unsung heroes supporting neurons. Among these, microglia are the brain's resident immune cells, constantly surveying their environment, ready to respond to injury or infection. Astrocytes also play a significant role, modulating the immune response and maintaining the delicate brain environment. Neuroinflammation is, at its core, the immune response within the brain and spinal cord. In its acute form, it is a vital protective mechanism. When the brain experiences an injury, infection, or trauma, microglia rapidly activate, migrating to the site of insult. They clear cellular debris, engulf pathogens, and release signaling molecules called cytokines that orchestrate a healing response. This acute inflammation is essential for recovery, helping to restore the brain to its healthy state. However, studies indicate that when this inflammatory response becomes chronic, persistent, or excessive, it can transition from being protective to being detrimental. This sustained neuroinflammation, often characterized by prolonged microglial activation and the continuous release of pro-inflammatory cytokines, can damage healthy neurons and impair synaptic function. Scientists have observed that chronic neuroinflammation is a common thread woven through many neurological and psychiatric conditions. For instance, research suggests that chronic neuroinflammation contributes significantly to the progression of neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease, where it exacerbates neuronal damage and accelerates cognitive decline. It is also increasingly implicated in the pathophysiology of mood disorders like depression and anxiety, as well as in chronic pain conditions. Factors contributing to chronic neuroinflammation are varied and often interconnected. They include chronic stress, as we discussed earlier, which can prime microglia to a more inflammatory state. Poor diet, particularly one high in refined sugars and unhealthy fats, can also fuel systemic inflammation that crosses into the brain. Infections, head trauma, and even genetic predispositions can all contribute to this silent, smouldering flame within the nervous system. The delicate balance of the brain's immune system is paramount. Understanding neuroinflammation helps us appreciate the intricate mechanisms that protect our most vital organ and the critical need to support a healthy environment within to keep these silent flames from raging out of control. It is a testament to the profound complexity of our nervous system that even its protective mechanisms require careful regulation to maintain harmony. Having explored the nervous system's responses to stress, its deep connection to our gut, and the delicate balance of neuroinflammation, we return to a concept that offers profound hope and underscores the dynamic nature of our being: neuroplasticity. We first encountered this remarkable capacity in previous chapter, recognizing the brain's ability to reorganize itself by forming new synaptic connections throughout life. Now, we delve deeper into its role in recovery, resilience, and overall well-being. Neuroplasticity is not merely about learning new facts. It is the fundamental mechanism by which our nervous system adapts, repairs, and even reinvents itself. Scientists have observed its power most dramatically in cases of neurological injury. Following a stroke, for instance, when certain brain areas are damaged, the brain doesn't simply give up. Instead, through intensive rehabilitation, it can recruit surrounding healthy brain regions to take over lost functions, forming new neural pathways to compensate for the damaged ones. This capacity for experience-dependent reorganization allows individuals to regain motor skills, speech, and cognitive abilities that were once thought to be permanently lost. This inherent ability to adapt extends beyond injury to shape our daily lives and enhance our well-being. Research consistently demonstrates that lifestyle factors can profoundly influence and enhance neuroplasticity. Regular physical exercise, for example, is not just good for the body, it stimulates the production of neurotrophic factors such as brain-derived neurotrophic factor, BDNF, which support the growth and survival of neurons and synapses. This means exercise can literally help our brains grow and stay flexible. Similarly, practices that cultivate mindfulness and meditation have been shown to induce structural changes in the brain, increasing grey matter density in regions associated with attention, emotion regulation, and self-awareness. A nutritious diet, rich in antioxidants and omega-3 fatty acids, provides the essential building blocks and protection necessary for optimal brain function and synaptic health. Even strong social connections and engaging in mentally stimulating activities contribute to a vibrant, adaptable nervous system, fostering what scientists call cognitive reserve, the brain's ability to cope with disease or damage by using alternative brain networks or more efficient cognitive strategies. The journey through the intricate patterns and subtle wisdom of the human nervous system is an ongoing exploration, a gentle current carrying us through vast and interconnected landscapes. We have witnessed its foundational elegance, its complex orchestration of our senses, movements, emotions, and thoughts, and now its profound capacity for resilience and renewal. As we conclude this chapter, we are reminded that our nervous system is a living testament to continuous adaptation, a symphony of our existence that constantly learns, heals, and evolves. We invite you to carry this sense of wonder and calm contemplation forward, allowing these insights to deepen your appreciation for the profound beauty and intricate dance within you. The journey continues, and the revelations, like gentle whispers, await. As we gently drift from the intricate dance of resilience and renewal, where the nervous system adeptly navigates stress and orchestrates its own healing, we find ourselves drawn deeper into the very architecture of thought, the profound landscape where consciousness itself emerges. We have explored the individual components, the neurons, the synapses, the specialized regions of the brain, each a marvel in its own right. Yet the true symphony of our existence, the richness of our inner world, arises not from these isolated elements, but from their vast and intricate interconnectedness. Tonight, we invite you to journey with us into the realm of neural networks. The exquisite tapestries woven by billions of neurons communicating in concert. Imagine not just individual threads, but entire patterns, constantly shifting and reconfiguring, giving rise to every thought, every feeling, every memory. Scientists have long understood that the brain does not operate as a collection of independent modules, but as a highly integrated system, where information flows through complex pathways, creating emergent properties far greater than the sum of their parts. We observe that a single neuron, though capable of remarkable feats of electrical signaling, is but a whisper in the grand chorus. Its true power is unleashed when it joins with thousands, even millions, of its counterparts, forming specialized circuits designed for specific tasks. These neural networks are the functional units of the brain, processing sensory input, driving motor commands, shaping our emotions, and enabling the highest forms of cognition. Research reveals that these networks are not static, they are dynamic, constantly adapting their connections and strengths based on experience, a testament to the enduring principle of neuroplasticity we have previously explored. From the simplest reflex arc to the most complex abstract reasoning, it is through these interconnected webs that the nervous system truly comes alive. As we contemplate the sheer scale of these connections, we encounter the fascinating concept of the Kegkonekam. Imagine a comprehensive map, an atlas detailing every single neuronal connection within an entire brain. While mapping the human connectome in its entirety remains one of the grandest challenges in neuroscience, scientists are making remarkable progress in mapping the connectomes of simpler organisms and even specific regions within the human brain. Current understanding suggests that the connectome is not just a static blueprint, but a dynamic entity, continuously rewired by learning, development, and even disease. Studies indicate that individual differences in our connectomes may underlie variations in personality, cognitive abilities, and even susceptibility to certain neurological conditions. It is a unique fingerprint of our neural self, a vast and intricate diagram of who we are, etched in the very wiring of our brains. We are learning that the strength and density of these connections, the specific pathways they forge, shape not only how we perceive the world, but how we interact with it, how we remember, and how we dream. Drifting further into this intricate landscape, we become aware of the brain's own internal rhythms, a subtle yet powerful orchestration of electrical activity known as brain oscillations, or brain waves. These rhythmic patterns, generated by the synchronized firing of vast populations of neurons, are not merely background noise. They are fundamental to how the brain processes information and coordinates its immense activity. We recognize distinct types of brain waves, each associated with different states of consciousness and cognitive function. Delta waves, 0.5 to 4 Hz, the slowest waves, predominant during deep, dreamless sleep. They reflect a state of profound rest and restoration, essential for physical and mental renewal. Theta waves, 4 to 8 Hz, often associated with light sleep, deep meditation, and states of creativity or daydreaming. They are also active during memory encoding and retrieval, providing a gateway to our subconscious mind. Alpha waves, 8 to 13 Hz. These emerge when we are in a relaxed yet awake state, often with our eyes closed. They signify a calm alertness, a peaceful readiness, and are frequently observed during states of quiet contemplation or meditation. Beta waves, 13 to 30 Hz, dominant when we are actively engaged in cognitive tasks, problem-solving, decision-making, or focused attention. They reflect a state of alert, active processing, often associated with external focus. Gamma waves, 30-100 plus Hz, the fastest waves, linked to higher-level cognitive functions such as perception, learning, memory, and consciousness. They are thought to bind information from different brain regions, creating a unified perception of reality, a true symphony of integration. Research reveals that the precise interplay and synchronization of these brain rhythms are crucial for healthy brain function. Disruptions in these oscillations are often observed in neurological and psychiatric disorders, highlighting their critical role in maintaining the delicate balance of our mental landscape. We find a profound beauty in this rhythmic dance, a silent, unseen ballet that underpins every moment of our conscious experience. As we continue our gentle exploration, we encounter a particularly intriguing neural network known as the default mode network, DMN. This network, comprising interconnected regions in the medial prefrontal cortex, posterior cingulate cortex, and angular gyrus, becomes most active when our minds are not focused on external tasks. Instead, it springs to life when we are daydreaming, reflecting on past events, planning for the future, or contemplating our own thoughts and feelings, essentially when we are resting our attention. The DMN is often referred to as the brains, to quote 25. but its activity is anything but idle. Scientists have observed its crucial role in self-referential thought, allowing us to construct our personal narratives, to ponder our identity, and to engage in social cognition, imagining the perspectives of others. It helps us consolidate memories, process emotions, and even fuels our creativity when our minds are allowed to wander freely. When we shift our focus to an external task, the DMN typically deactivates, allowing task-positive networks to take over. The delicate balance between the DMN and these task-positive networks is vital for cognitive flexibility and mental well-being. An overactive or underactive DMN has been implicated in conditions ranging from depression and anxiety to Alzheimer's disease, suggesting its profound influence on our internal landscape. We recognize its quiet hum as the background music of our inner world, always playing even when we are not consciously listening. This journey into neural networks and brain rhythms naturally leads us to one of the most profound and enduring mysteries of the nervous system: consciousness. How does the intricate dance of billions of neurons, the synchronized firing of electrical impulses, give rise to our subjective experience, the feeling of being, the awareness of self, the vividness of our perceptions, the richness of our inner thoughts? This is often called the hard problem of consciousness, a question that continues to captivate and challenge scientists and philosophers alike. While we may not yet possess a complete answer, current understanding suggests that consciousness is not localized to a single brain region. Instead, it is believed to be an emergent property of highly integrated and complex neural activity across multiple brain networks. Research points to the neural correlates of consciousness, NCCs, the minimal set of neuronal events and mechanisms sufficient for a specific conscious percept. We observe that consciousness often involves widespread communication across the brain, particularly involving the thalamus and cortical areas, where information from various sensory modalities and cognitive processes is unified into a coherent, subjective experience. Theories like the global workspace theory propose that consciousness arises when information becomes globally available to many different brain systems, allowing for flexible access and processing. Integrated information theory suggests that consciousness is related to the amount of integrated information a system can generate, emphasizing the interconnectedness and differentiation of brain activity. We recognize that our sense of self, our unique identity, is inextricably linked to these complex neural networks and their ongoing activity. The continuous stream of information processing, memory retrieval, emotional responses, and future planning, all orchestrated by these interconnected systems, forms the bedrock of our personal narrative. It is within this vast and dynamic neural architecture that our individual consciousness takes shape, allowing us to experience the world, to reflect upon it, and to know ourselves as distinct beings. The profound beauty lies not just in the existence of these mechanisms, but in their capacity to create the very fabric of our subjective reality, transforming electrochemical signals into the vibrant tapestry of our lives. As we gently bring our contemplation to a close, we are left with a renewed sense of wonder at the incredible complexity and elegance of the human nervous system, from the quiet hum of the default mode network to the synchronized rhythm of gamma waves, from the intricate wiring of the connectome to the profound mystery of consciousness. we are constantly reminded that within each of us lies a universe of unparalleled intricacy. This journey has shown us that the brain is not merely a collection of parts, but a dynamic, self-organizing system, constantly adapting, learning, and creating the very essence of who we are. We invite you to carry this sense of peaceful curiosity forward, knowing that the exploration of our inner world is an endless, awe-inspiring adventure. As the gentle current of our exploration carries us forward, we find ourselves standing at the precipice of a profound realization. The human nervous system is not merely a receiver of information, but a masterful architect, constantly constructing the very reality we inhabit. The previous chapter left us contemplating the vast, intricate dance of neural networks and the enigmatic nature of consciousness itself, how the integrated symphony of our brain gives rise to subjective experience. Now, we delve deeper into this active construction, exploring how our perceptions are shaped, how our sense of self emerges, and how we connect with the complex tapestry of other minds. We invite you to drift with us into this tranquil space of understanding, allowing the subtle wisdom of your own being to unfold before you. As you settle into a state of calm contemplation, let your curiosity be a gentle guide through the intricate patterns that define our inner and outer worlds. Our journey begins with the fundamental understanding that what we perceive is not a direct, unmediated reflection of the external world, but rather an elaborate, dynamic interpretation crafted by our brain. Scientists have long observed that our sensory organs gather raw data, yet it is the sophisticated processing within our nervous system that transforms these signals into the rich, meaningful experiences we call reality. Consider for a moment the phenomenon of the blind spot. Within each of our eyes there is an area on the retina where the optic nerve connects, containing no photoreceptors. Logically, this should create a noticeable gap in our visual field, yet we rarely, if ever, perceive a hole. This is because our brain actively fills in this missing information, seamlessly constructing a continuous visual scene based on surrounding details and information from the other eye. Research indicates this is a testament to the brain's proactive role, demonstrating its capacity to predict, interpolate, and present a coherent, albeit constructed, reality. This active construction extends far beyond simply filling in gaps. It is deeply influenced by top-down processing, where our expectations, prior knowledge, memories, and emotional states profoundly shape what we perceive. Imagine walking through a forest. If you're expecting to see a specific type of bird, your brain might be primed to notice subtle movements or colors that you would otherwise overlook. Studies in cognitive neuroscience highlight how our past experiences create perceptual frameworks, guiding our attention and interpreting ambiguous sensory input. This means that two individuals can experience the exact same external stimulus, yet perceive it differently based on their unique internal landscapes. Furthermore, our nervous system masterfully employs sensory adaptation, a remarkable ability to tune out constant, unchanging stimuli. When you first put on a watch or feel the fabric of your clothes, you notice it. But within moments, your sensory neurons, specifically those involved in touch, reduce their firing rate in response to the continuous input. This allows our attention to be freed from redundant information, preserving cognitive resources for novel or important changes in the environment. It is a subtle, yet profound act of filtering, ensuring that our awareness remains focused on what truly matters. In some rare and fascinating instances, this sensory construction takes an extraordinary turn, leading to phenomena like synesthesia. For individuals with synesthesia, stimulation of one sensory or cognitive pathway leads to automatic involuntary experiences in a second sensory or cognitive pathway. For example, some may, again to quote 29, colours when they hear music, chromesthesia, or, quote 30, shapes, or resta, quote 31 to dine textures when they read words. Current understanding suggests that synesthesia arises from a typical, heightened cross-activation between different brain regions that are typically more segregated. It offers a vivid glimpse into the brain's capacity for interconnected, multimodal construction of experience, reminding us of the fluid boundaries within our perceptual architecture. From the grand architecture of perception, we gently turn our focus inward towards one of the most profound creations of the nervous system, our continuous evolving sense of self and identity. How does a collection of neurons, synapses, and electrical impulses give rise to the unique I that experiences, remembers, and plans? This question has captivated philosophers and scientists for centuries, and while a complete answer remains elusive, neuroscience offers tantalizing insights into its neural underpinnings. Our sense of self is not a static entity, but a dynamic narrative, woven together by countless threads of experience and memory. A cornerstone of this narrative is episodic memory, the ability to recall specific events from our personal past, complete with their context of time and place. Research shows that the hippocampus, crucial for forming new episodic memories, works in concert with the prefrontal cortex and other cortical areas to bind these experiences into a coherent, autobiographical timeline. It is through this continuous stream of remembering and re-experiencing our past that we maintain a sense of who we have been, grounding our present identity. The prefrontal cortex, often considered the seat of executive functions, also plays a pivotal role in shaping our personality, self-awareness, and our ability to reflect on our own thoughts and actions. This region is critical for self-regulation, decision-making, and understanding social rules, all components that contribute to our unique persona. Damage to the prefrontal cortex, as observed in various neurological conditions, can profoundly alter an individual's personality and sense of self, underscoring its indispensable contribution. Furthermore, our very physical presence in the world contributes to our sense of self. Body schema and proprioception. The brain's continuous unconscious map of our body's position, movement, and orientation in space are fundamental. Sensory input from muscles, tendons, and joints, processed in the somatosensory cortex, creates a constant, updated internal model of our physical self. This internal map grounds our experience, allowing us to feel quote, 33 in our own skin and distinguishing our body from the external world. Studies involving virtual reality and prosthetic limbs reveal the remarkable plasticity of this body schema, demonstrating how our brain can integrate new sensory information to extend or alter our perceived self. And then there is the default mode network, DMN, which we touched upon in the previous chapter. This network of interconnected brain regions consistently shows increased activity when we are not focused on the outside world, but are instead engaged in internal thought processes such as mind wandering, envisioning the future, recalling memories, and, significantly, reflecting on ourselves and others. Scientists theorize that the DMN is intimately involved in constructing and maintaining our self-narrative, allowing us to process personal experiences, consolidate our identity, and even simulate social interactions. Its constant hum beneath the surface of our focused attention reminds us that our sense of self is an ongoing, active construction, even in moments of rest. Having explored the intricate construction of individual perception and self, our journey now expands to encompass the profound ways our nervous system allows us to connect with others, to understand their intentions, and to share in their emotional landscapes. This capacity for social cognition, for empathy, is a hallmark of human experience, and its neural foundations are a testament to the nervous system's sophisticated design. At the heart of our ability to understand and even feel what others are experiencing lies the remarkable mirror neuron system. First identified in primates and later strongly implicated in humans, mirror neurons are a class of visuomotor neurons that fire both when an individual performs an action and when they observe the same action performed by another. For instance, the same neurons might activate when you reach for a cup and when you watch someone else reach for a cup. Research suggests this system is crucial for imitation, learning, and, significantly, for understanding the intentions behind others' actions and empathizing with their emotions. It provides a neural mechanism for, quote 35 under, the experiences of others within our own minds, blurring the lines between observer and observed. Building upon this foundational mirroring, our nervous system allows us to develop theory of mind, TUM, the cognitive ability to attribute mental states, beliefs, intentions, desires, knowledge, to ourselves and others, and to understand that these mental states can differ from our own and from reality. This complex skill, typically developing in early childhood, is essential for successful social interaction, allowing us to predict and explain others' behavior. Neuroimaging studies have identified several key brain regions involved in TOM, including the temporoparietal junction, TPJ, which is critical for distinguishing between self and other perspectives, and the medial prefrontal cortex, MPFC, involved in reasoning about mental states. These regions form a sophisticated network that enables us to navigate the intricate social world. The very fabric of our social bonds is also deeply influenced by specific neurochemicals. Oxytocin, often referred to as the love hormone or bonding hormone, plays a crucial role in promoting social attachment, trust, and empathy. Released during positive social interactions such as touch, eye contact, and intimacy, oxytocin acts on receptors throughout the brain, enhancing feelings of connection and reducing social anxiety. Alongside oxytocin, vasopressin also contributes to social behavior, particularly in pair bonding and paternal care. The intricate interplay of these neurochemicals highlights the biological underpinnings of our deepest social connections, demonstrating how profoundly our nervous system is wired for communal existence. As we gently conclude this chapter, we are left with an even deeper appreciation for the nervous system's unparalleled complexity and its profound beauty. From constructing our perception of reality to weaving the narrative of ourself, and then extending that understanding to connect with others, the brain is an extraordinary orchestrator of experience. It is a testament to an intricate design that allows us not only to exist, but to thrive in a world of shared meaning and connection. We invite you now to let these insights settle, to appreciate the quiet miracle within. As the day's light gently recedes, we prepare for further discoveries, allowing the gentle current of knowledge to carry us toward a deeper, more peaceful understanding of our own remarkable existence. As we gently drift further into the intricate landscape of our nervous system, we have journeyed through its foundational structures, its chemical dialogues, its sensory perceptions, and its profound capacity for social connection. We have witnessed the brain as a master constructor of reality, shaping our very sense of self and our interactions with the world around us. Now we invite you to turn your gaze inward once more, to contemplate the nervous system's most extraordinary attribute, its ceaseless capacity for change, growth, and adaptation throughout the entire span of our lives. We touched upon neuroplasticity earlier, acknowledging its role in learning and recovery. But let us now settle into a deeper appreciation of this phenomenon, for it is not merely a feature, but the very essence of our nervous system's dynamic existence. Research consistently reveals that our neural pathways are never truly static. They are constantly being refined, reshaped, and rewired in response to every experience, every thought, every emotion, and every interaction. From the first moments of development to our final days, this intricate network is a living, breathing testament to adaptation. At the heart of this remarkable adaptability lies synaptic plasticity, the ability of the connections between neurons, the synapses, to strengthen or weaken over time. When we learn something new, when we practice a skill, or when we repeatedly experience a particular sensation, certain synaptic connections are enhanced. This process, often referred to as long-term potentiation, LTP, makes it easier for neurons to communicate with each other in the future, forming the cellular basis of memory and learning. Conversely, connections that are rarely used can weaken through long-term depression, LTD, a crucial mechanism for pruning away irrelevant information and maintaining neural efficiency. Imagine a vast interconnected forest, where paths are either deepened by frequent travel or slowly reclaimed by nature when left unused. This continuous sculpting of neural architecture allows us to learn, to grow, and to continually integrate new information into our understanding of the world. This profound capacity for change is intimately linked to the processes of learning and memory. Our nervous system is a relentless archivist, constantly encoding, consolidating, and retrieving information. When we encounter new information, our brain engages in encoding, transforming sensory input into a form that can be stored. This initial encoding often involves the hippocampus, which acts like a temporary staging ground for new memories. From there, through processes that scientists are still unraveling, these nascent memories undergo consolidation, a more permanent storage process that often involves the transfer of information to various areas of the cerebral cortex for long-term retention. And finally, when we need to recall a piece of information, our brain performs retrieval, accessing and reactivating those stored neural patterns. The tapestry of our memory is incredibly rich and varied, extending far beyond the explicit and implicit distinctions we explored previously. We possess Semantic memory, our storehouse of general knowledge and facts about the world, the capital of France, the laws of physics, the meaning of words. Then there is episodic memory, our autobiographical record of specific events and experiences imbued with the context of time and place. Your last birthday party, the first time you rode a bicycle, the scent of rain after a dry spell. These memories give richness to our personal narrative and contribute significantly to our sense of self. Furthermore, procedural memory allows us to perform learned skills and habits automatically, without conscious effort, riding a bike, typing on a keyboard, playing a musical instrument. And let us not forget working memory, a vital temporary storage system that holds and manipulates information for brief periods, allowing us to follow a conversation, solve a problem, or remember a phone number just long enough to dial it. Each type of memory relies on distinct yet interconnected neural circuits, weaving together the intricate fabric of our past and present. A truly remarkable aspect of memory consolidation unfolds during sleep. As we surrender to the nightly embrace of slumber, our brain doesn't simply shut down. It enters a highly active state of memory reprocessing. During deep non-REM sleep, the brain appears to replay recent experiences, transferring newly acquired information from the hippocampus to more stable, long-term storage sites in the cortex. REM sleep, with its vivid dreams, also plays a crucial role in consolidating emotional memories and integrating new information with existing knowledge. This nocturnal work underscores the profound importance of restful sleep, not just for physical restoration, but for the very architecture of our memories and our capacity for future learning. This journey of adaptation begins long before we are consciously aware. The development of the nervous system is a breathtaking ballet of genetic programming and environmental influence. From the formation of the neural tube in the earlier stages of embryonic development, billions of neurons are born through neurogenesis, migrating to their destined locations and forming trillions of connections. In childhood and adolescence, a process called synaptic pruning occurs, where unused or inefficient connections are eliminated, refining the neural network and making it more efficient. Simultaneously, myelination, the insulation of axons with fatty myelin sheaths, accelerates neural signal transmission, allowing for faster and more complex thought processes. This period is also characterized by critical periods, specific windows of time during which the brain is particularly receptive to certain types of environmental input, such as language acquisition or visual development. The environment we inhabit, the interactions we have, and the stimuli we encounter profoundly shape the developing brain, laying the groundwork for who we become. Yet, this dynamic transformation does not cease with maturity. The nervous system continues its remarkable dance through the just brain. While it is true that certain cognitive functions may experience a gradual decline with age, such as processing speed or aspects of working memory, the aging brain also demonstrates remarkable resilience and adaptability. Scientists speak of cognitive reserve, the brain's ability to cope with age-related changes or even pathology by utilizing alternative neural networks or more efficient cognitive strategies. This reserve is often built up through lifelong learning, engaging in mentally stimulating activities, and maintaining social connections. We understand now that healthy aging is not about preventing change, but about fostering the brain's capacity to adapt and thrive. Lifestyle factors such as regular physical exercise, a balanced diet, stress management, and continued intellectual engagement are powerful allies in supporting brain health and promoting graceful cognitive aging, even enabling some degree of continued neurogenesis in areas like the hippocampus. And what of the nervous system's capacity for healing and regeneration? For a long time, it was believed that once neural tissue was damaged, it was lost forever. While the central nervous system's regenerative capabilities are indeed limited compared to other tissues, current research reveals a more nuanced and hopeful picture. We know that neurogenesis, the birth of new neurons, continues throughout adulthood in specific regions, notably the hippocampus, important for memory and learning, and the olfactory bulb, involved in smell. While extensive regeneration of damaged axons in the spinal cord or brain remains a significant challenge, the brain's incredible plasticity allows for remarkable recovery through adaptive plasticity. Following injury, such as a stroke, the brain can reorganize its remaining healthy tissue, forming new connections and recruiting different areas to compensate for lost function. This is the principle behind rehabilitation therapies, where repeated practice and focused effort can guide the brain to rewire itself, allowing individuals to regain lost motor skills or cognitive abilities. Glial cells, once thought of as mere support staff, are now understood to play crucial roles in this repair process, clearing debris, guiding new growth, and modulating inflammation. As we conclude this chapter, we are left with a profound sense of awe for the nervous system's journey through time, from its nascent formation to its continuous evolution, through learning, aging, and even healing. It is a testament to an ever-changing, ever-adapting marvel, constantly striving for balance and optimal function. The future of neuroscience promises even deeper insights into these processes, with research frontiers exploring advanced neuroimaging, targeted therapies for neurological disorders, and the potential of brain-computer interfaces to restore function and enhance human capabilities. We invite you to carry this understanding of the nervous system's dynamic nature into your quiet contemplation. Recognize that your brain is not a fixed entity, but a living, breathing tapestry woven anew with each passing moment. This realization can be a source of profound peace and empowerment, knowing that within you resides an unparalleled capacity for growth, learning, and resilience. Allow this gentle truth to settle within you, enriching your calm contemplation as you continue your journey into peaceful rest.